Hi everyone, welcome back to our topic 5 video. Now we're going to look into 5.1 nutrients, which actually covers 4 learning objectives, but today we're going to look at 2 learning objectives. The first one is 5A, which is list the chemical elements that make up carbohydrates, fats, and protein. And 5B, describe tests for starch, which is the iodine in potassium iodide solution, reducing sugars using Benedict's solution, and also protein using the Bio-Rat test. Reducing sugar is not protein, by the way. I need to edit that later. And we're also going to look at fats as well. So actually protein is a separate test. So I'm gonna have to do this. Yep. Give it that, and we're good. Alright. So the last test is the fat ethanol emulsion test, and we're going to look at it later. So if you've watched the first video, then you would know that we have talked about the chemical elements that make up protein. The step to success today is, if you can, you should be able to list out and try to list out all the elements that are found in carbohydrates, fats, and protein. So if you actually have your Cornell notes ready, that'll be really good for you to do. Make sure that you have this inside your notes. Number 2, write down step by step the test for carbohydrates, proteins, and fats, as well as the expected observations and also their conclusions for both the positive and negative results. So now let's look into the elements that are found in carbohydrates. Carbohydrates are molecules, and they are biological molecules that are made up of these elements: carbon, hydrogen, and oxygen. Carbohydrates actually mean carbon and water, so that's why it's called carbohydrate. So it's made out of carbon, and it's not made out of water, but this water part, the hydrate part, and carbohydrate actually comes from the fact that hydrogen and oxygen atoms are present in the ratio of 2:1, just like in water. So the types of carbohydrates that we have, number 1, they are called monosaccharides. So just a fact here. Mono here means 1, and saccharides, this word saccharide actually means sugar or sweet. So monosaccharides are the sweetest of all the carbohydrates. There are 3 examples. Number 1, which is sugar. Second, it's fructose. Third is galactose. Number 2, the second category, is called disaccharides. And di here actually means 2. Under disaccharides, we have maltose, sucrose, and lactose. And the third category is polysaccharide. Poly doesn't mean 3, but it actually means many. And you have 3 examples here too. The first one is starch, second one is glycogen, and the third one is cellulose. So all of these words, we are going to be mainly hearing about glucose. Maybe sometimes we'll learn about or talk about sucrose and lactose. We'll also talk a lot about starch, sometimes glycogen, and also a lot of cellulose as well. So these 3 are pretty important. Monosaccharides actually mean 1 and single. So it means they're a single sugar molecule. So they are all made up of single sugar molecule. For example, here we have fructose, which can be found in fruits, and galactose, which can actually be found in milk. So this is the type of sugar, so simple sugar that you can find. Another simple sugar is actually the disaccharides, and it's made up of 2 units of sugar. You have your maltose, and then you have, here is lactose, which I think you're very familiar with, which also comes from milk. And actually you should know sucrose too, because sucrose is a type of disaccharide, the type of sugar they can find in plants. So sugar cane, the type of sugar that you find in your pantry, in the tabletop, wherever your kitchen is, that is sucrose. And polysaccharides like starch, this is how starch actually looks like. It's made up of a lot of these is it a pentagon or hexagon? I need to Google that. But basically it's made up of a lot of sugar units here. So there's not just one or two, there's actually many. So that's an example of starch where you should know your sources of starch, and that's a dancing potato. And you also have glycogen. So glycogen is the type of polysaccharide that you actually store in your liver and in your muscle cells. So when you actually eat a lot of starch, you're going to break it down to maltose first and then glucose. This glucose is the one that will be transported to all parts of your body in your blood. So once your blood sends the glucose into your liver or your muscles, if you have excess, you're going to first store it as glycogen, and then if you have even more excess, then it will be converted to fat. So the third one here, this polysaccharide is cellulose, and you should know a little bit about them because you have learned something about them actually. You can find these a lot in your veggies and in plants. So if you remember your structure of a plant cell, the plant cell has a cell wall. So that cell wall is actually made up of a polysaccharide, a carbohydrate called cellulose. Next, we're going to look at the elements that are found in fats. Fat molecules are also biological molecules, and they're made up of the elements carbon, hydrogen, and oxygen. So it's actually the same as carbohydrates. But unlike carbohydrates, fat contain much less oxygen in proportion to hydrogen. So we're here talking about the ratio. Just now we know that carbohydrate means carbon and hydrate, so like water. So the ratio of hydrogen to oxygen is 2:1. But for fat, it's much less oxygen in proportion to hydrogen. So let's see a comparison between the two. So you have these two classic examples of carbohydrate and fat. So just so you know, your honey there contains half of them, I think, contains glucose and half fructose. And you have right there steak and a lot of butter sizzling hot. And here you can find this type of fat called triglycerin, which is beef fat. So glucose has a molecular formula of C6H12O6. So if you actually check the ratio here, it's 2:1 as well for hydrogen and oxygen. For triglycerin, you have C57, H110, and O6. You can see that the oxygen is much more less than the hydrogen. This is 2:1, but this is like much more lower right here. So look at those two right there, and you can see that is the main difference between carbohydrates and fat. Oh, I'm sorry. Why is this here? Okay, this shouldn't be here. Okie dokie. Alright, so I think this is a summary of everything. What are the elements that are found in protein, starch, and fat? So one fact that you need to know is that all biological molecules always contain carbon and hydrogen, but not all biological molecules are the same. That's why they have different names. Protein molecules, for example, contain the elements carbon, hydrogen, oxygen, nitrogen, and sulfur, whereas carbohydrates and fat molecules, they contain the elements carbon, hydrogen, and oxygen. You can see that both protein and carbohydrate and fat, the third one here, they both contain carbon and hydrogen elements. Alright, so now that we know about the elements that are found in those food nutrient categories, we need to also learn about how to test for them. Why do we test for food? Some of the most important reasons for testing food, it's not about the taste, but more like, is it legit? So when you want to give people food, you need to make sure that it is very safe. So food test actually has that main reason. We need to test for the food, whether or not it contains all the things that we think it contains. So the 3 food tests that you will need to learn in O-levels is number 1, to test for carbohydrates. Remember that carbohydrates, you have 3 types. You have your monosaccharides, the simple sugars, and your disaccharide, which is also simple sugar. And you also have your polysaccharide, which is starch, cellulose, and glycogen. But for the food test in O-levels, you're going to learn about the polysaccharide, testing for starch using iodine solution, and you're also going to learn about how to test for reducing sugars. So when we talk about reducing sugars, we're actually talking about simple sugars, and we're going to use this solution called the Benedict solution. Sometimes they're called Benedict's reagent as well. Second group of food we're going to test is protein, and this is called the biuret reagent that can actually help us identify if there's protein present in the food sample. The third type of test is food test for fat, and the type of food test here is called the ethanol emulsion test, and you're actually going to use ethanol solution here. So let's look at how to test for starch. First thing you need to do is you need to add a few drops of iodine solution directly onto your food sample, and then you can observe for a color change. So the solution we're using here is iodine. For solid samples, you'll need your white tile. You'll also need to place your food sample on your white tile, and you need to add a few drops of iodine solution to it. Note that iodine originally is actually brown, or called yellow-brown in color. For liquid samples, you're going to need your test tube. You're also going to need to drop the iodine solution into your food sample. But don't forget to put the food sample inside the test tube first, and then you add your iodine. So these are 2 examples of results that you will actually see. So this is a piece of bread, and you can see that after adding the iodine solution, it has this kind of black color. We actually call that the blue-black color. So blue-black color means that it is a positive test. That means that there is starch present. So these are 2 examples of how liquid tests may look like. Here you have a positive test where you see that blue-black color, and this is a negative test. It means that the solution remained yellow-brown, and that is the color of iodine solution. So the observation and conclusion. So observation for a positive test, you should see a blue-black color was observed, or you can say the solution turned, or there is a color change from yellow-brown to blue-black. Actually, that's even better than just saying that. So let me just move this up, and I'm going to move this down. Okay, so it's better to change it to this. So when you're writing this down at your practical exams, make sure to say what color that it started off with and what color did it end with. So the positive test right here is that there will be a color change from yellow-brown to blue-black observed, and the interpretation or the conclusion here is that starch is actually present. But that's not an observation, yeah? This is what we can conclude from the observation. Observations are usually to do with what color can you see, or like what precipitate can you see. So if you get like a negative test, you will see that the brown color, of the iodine solution remained and there's no color change observed. This is when you have starch being not present. So this is also an interpretation from the observation that you see. Next is reducing sugars. Before I talk about how to test for them, I think I need to tell you guys what are reducing sugars. Reducing sugars are actually simple sugars like monosaccharides and disaccharides, except for one, which is sucrose. So you have your monosaccharides, which is your glucose, fructose, and your galactose, and you also have your disaccharides, which is maltose, sucrose, and lactose. So monosaccharides are made up of one unit of sugar molecule. You can find this in your fruits and also milk. And glucose can actually be found in basically everything, including your potatoes. And you can also find these things called disaccharides, which is also simple sugar. It's sweet as well. And lactose is found in milk, and sucrose is found in plants. But maltose is actually a plant substance as well. So just to clarify with you guys, all of these except for all of these except for sucrose is a reducing sugar. So reducing sugars are all simple sugars, monosaccharides, disaccharides, except for sucrose. There's a reason for this, and I think I'll explain to you that in another video. Okay, so now that we know our reducing sugar. So the first thing we need to do is actually we need to place 2 cm³ of the sample into a test tube. Okay, I actually made a flow of this, so I need to make sure that that doesn't come out first. Okay, let's restart that. So how do we test for reducing sugars? We don't use iodine, we use Benedict's. Benedict''. I think it's called Benedict's because a person called Benedict is the one who actually created this test. So that's just my guess, but you can Google that. Okay, so the first thing we need to do is to place 2 cm³ of the sample into a test tube. And then you add 2 cm³ of Benedict's solution to the food sample, and then you shake the mixture well. So if you have a solid sample, you need to cut it up first. And if you can mash it up, that'll be really good, but as long as it's in tiny pieces, that'll be great. After that, you put those tiny little pieces, don't say tiny pieces, just say crushed or cut food samples into a test tube, a clean test tube, and you add equal volume. So if it's a solid, don't forget to actually add distilled water. After you add distilled water, you should shake that mixture. And why we need to shake it is actually so that the reducing sugar, so the small sugar that can be found inside the food sample, will actually dissolve in the distilled water. So this is the thing that you're going to get. You're going to get those food samples, and you're going to get a bit of the food sample in the water, and you're going to add the equal volume. Usually we use 2 cm³ of Benedict's. How do we measure 2 cm³? You can measure it if you actually have a measuring cylinder, but if you don't, you can just kind of eyeball that and just like use your finger or like a ruler to kind kind of just estimate 2 cm cube. Okay, so once you have all of this, so once you've added 2 cm cube of Benedict solution to the food sample and the dissolved fluid sample, you should shake the mixture very well even more. So let me just change that heading. Copy and oopsies. Okay, paste right there. Gonna move my cat to the side. Gonna move my instruments to the side, and then we're good. Okay, so after you have shaken your sample, you need to place the test tube into boiling water bath for 2 to 3 minutes, and then you have to observe for a color change. So what this means is that you add those two, make sure to shake it, okay? Don't have them like in separates like this. So make sure that there's water in there, and then you have to heat that up. So you have to heat that up in a water bath for around 2 to 3 minutes, or even about 5 minutes, but not more than that. Otherwise, you might burn your sample. So after you place it in that water bath for 2 to 3 minutes, you're going to observe a color change. If you see that your solution remained blue, this means that there's no reducing sugar present. If your sample looks like this, like green or yellow, so you have precipitate as well. PBT here means precipitate. That means there's traces of reducing sugar. It means that there's a little bit there. If it turned orange, that means there's moderate amount of reducing sugar. Remember to use the word reducing sugar because we don't want to say sugars. Sugars here mean that you're including sucrose, and sucrose is not a reducing sugar. If your sample, you can observe this brick red precipitate, that means that there's a large amount of reducing sugar in your sample. So what kind of observation and conclusions can we actually get? So if it's a positive result, you will see or observe blue, green, not blue, okay? Blue, green. So we're talking about, I think this one, that's kind of like bluish green, but to me that's just green. Anyway, blue, green, but not blue. So green, yellow, orange, brown, red or brick red precipitate is formed. So not just a color change, but also mention there's precipitate, because if you actually look at that sample for real, you can see that there is some precipitate present and formed. And the interpretation here is that when you see this observation, it means that reducing sugar is present. But if you have a negative result, what will it look like? The mixture just remains blue. So just say that it just remains blue because there's no color change there. Reducing sugar then therefore is not present. So let's just look at that picture again. This is how Benedict solution looks like. And if it is negative, this is how it remains blue. Okay, and now we're moving on to testing for proteins. So the first thing you need to do is you need to place 2 cm cube of, okay, not sodium hydroxide actually. Let's just say sample. To a test tube. Alright, so when you have Bio-Ret reagent, you are not going to use sodium hydroxide. That is only if you don't have Bio-Ret reagent, but I will not focus on that today because in your learning objective, you will be asked about Bio-Ret reagent instead of the other one. So let's look at Bio-Ret reagent testing for proteins today. So the first thing you need to do is you need to place 2 cm cube of your sample into a clean test tube. And then you should add equal volume of Bio-Red reagent and shake it and allow it to stand for 5 minutes. And then you should observe for a color change. So this is an example for liquid samples, but again, if you have solid samples, make sure to cut it first. Don't forget your white tile and your scalpel. And then after that, you need to add the distilled water. You shake it so that you can dissolve the nutrients into that distilled water. And then finally, you can move on to this part. So make sure that it's 2 cm cube of that volume of distilled water and also the crushed food sample or the cut food sample, and you need to add equal volume of Bio-Red reagent or Bio-Red solution. So you need to have 2 cm cube of Bio-Red and 2 cm cube of your sample. And then you just shake it and allow it to stand. So how do you make your test tube stand? You put it on a test tube rack. So you'll wait for 5 minutes and see if there's any color changes. Note that Bio-Red solution is originally blue in color as well. So if you have a negative result, it's going to remain blue, but if it's a positive result, then it will actually turn purple. So the observation is that if the solution changed from blue to purple, that is a positive result, and that means the interpretation here is that reducing sugar is present. But if the solution remains blue in color, that means that, oh my gosh, not reducing sugar, we're looking at protein here. Sorry, protein is present and protein is not present. Okay, so because we're looking at Bio-Red test for protein, the interpretation should be protein is present if it is from blue to purple, and if the solution remained blue, then that means that protein is not present. I need to check this as well. Did I write down that? Okay, cool. I did. Right. So here we go. Test for fat. So the first thing you have to do is to cut and crush the sample to be tested and place it in an empty test tube. This is a little bit different, and this is where a lot of people get wrong, is this part right here, which is you should add 2 cm cube of ethanol to the solution to be tested, and then you shake it very, very thoroughly. You don't put in water. This time you put in ethanol instead. Why ethanol? This is because fat, or aka lipids, that's the same thing. We mainly use lipids, but sometimes we use fat. Both of those things, those nutrients, those biochemicals, they dissolve in ethanol, so alcohol instead of water. So what you need to do is get your white towel. Usually you'll be given peanuts. Peanuts? Is that? No, peanuts for protein. No, actually, yeah, peanuts for fat test. And then you would actually need to cut those peanuts up into little pieces. Then make sure to put the peanut into the test tube, the little pieces, and then add ethanol, not water, okay? Ethanol. So after you add ethanol, 2 cm cube of it, and then you're going to shake it vigorously until that food sample, the fat in there, is dissolved in the ethanol. Now the thing is that it's a little bit special as well, and the fact that you kind of don't want the pieces of peanut to be there. So what you need to do is you need to decant that mixture. So decanting just means you transfer the liquid part into a new test tube. And then finally you add water. So you only add water at the end of the fat emulsion test. And then you can observe if there's any changes. So remember, the first thing you need to do is to decant. After you mix it steady with ethanol, you decant. So you remove, or you just transfer the liquid part. So what you're going to get, sorry about that, So let me just change the animations for this. You can see how much effort that this actually requires. Okay. Okay. So I'm going to delete. So this is also a tutorial on how to make presentation slides. Let me just see. This one, they appear. Okay, I'm gonna click on this little guy. Appear. Appear. I think you can actually guess what happens next. So, alright. Okay, let's rewind again. Oh no, I forgot about that. Okay, I think I need to move that there. Here. Okie dokie. I think we're good. Alright, now back to business. Okay, I think we need to go back here. Okay, so after we've cut our peanut into little pieces, we put them in a test tube, add the ethanol to dissolve the fat or lipid in the peanut, so we get 2 cm cube of the sample. Then after that, we want to decant. So decanting means to remove the liquid, to transfer the liquid into a new test tube. After decanting, you're going to only get the clear, not really clear, but just it doesn't have any solids in there. So next, what you need to do finally is to actually add distilled water. So when you add distilled water, at the end, you can now observe for any changes. So the kind of observation that you will see is these two. So this is the negative test where the liquid here is just remaining clear, crystal clear. However, if you have a positive, so that means that fat is present, you're going to see an emulsion. So this is why we call it the ethanol emulsion test. So we call this that there is either white precipitate, you can say, or it is cloudy white suspension. So for the observation here, solution will remain colorless and no emulsion is formed. So lipids are not present. But if you see a layer of cloudy white suspension that forms either at the top of the solution or like throughout, then that means that lipids are present. So upon close inspection, if you actually have a look when we are able to do this experiment, there are actually tiny globules of fat suspended at the solution that can be seen. This is why we call it an emulsion. So food with high fat, aka lipid content, will have higher layer than foods with less. So this means that with this test, ethanol emulsion test, you can even measure if that food sample has high fat lipid or lower content by looking at how much fat is suspended at the top. So that emulsion, that is a cloudy white emulsion that is formed. And that is all for today, guys. So this is the end of the second part of 5.1 nutrients, and I'll see you guys next time.